Thank you for distinguished guests and speakers. And thank you all for participating in our panel discussion today. As the moderator just introduced, our topic of focus will be cell and gene therapy, which is a completely new pharmaceutical modality that emerged for the past decade. And within the past decade, cell and gene therapy has achieved not only clinical POC, but also a market success. Some of the cell and gene therapy products is enjoying a tremendous market commercial success, actually. And for cell and gene therapy, we can categorize into maybe 4 different categories. First is in gene replacement therapy. That's what our company is pursuing. And the second is gene editing, which is Dr. Tian's company is pursuing. And the third one is cell-based therapy, which is Yarong's company is pursuing. And lastly is a RNAi and mRNA, so RNA-based gene therapy, which is Shiyu's company is pursuing right now. So today we're going to focus on the discussion on 4 different topics. First is efficacy of cell and gene therapy. Second is the safety of cell and gene therapy. Third one is going to be the cost of cell and gene therapy product. And lastly is definitely we're talking about the recently explosion of BD opportunities globally. So first I would like to start by introduction of our guests and also about each individual company. May we again start from Shiyu.
This work? Yeah, okay. Yeah, it's a great pleasure to participate in today's panel discussion. And then thank you, Zhenhua, for the introduction. And actually, Zhenhua has been a really great friend and then a very nice, supportive advisor along the way that we found SaneGene. Thank you, Zhenhua. So this is Shiyu Wang and from SanegeneBio and then CSO and co-founder. SaneGene was founded 3 years back with 3 financing rounds completed and then totally about 330 million as a total. And then SaneGene positioned ourselves at Boston, Shanghai, and Suzhou. And the company, as Zhenhua just mentioned and introduced, and is really focused on small interferon nucleic acid therapeutics.
Go ahead.
My name is Shin-Shay Tian. I'm really happy to be here today. Yeah, I'm the head of discovery biology at Shanghai Vitalgen. And Vitalgen is based in Shanghai, as you can see from the name. Yeah, and we are a clinical stage gene therapy company. We actually also work on gene replacement therapy. And actually, our clinical stage products are all gene replacement therapy. And we are working very hard to advance our gene editing projects into clinics. Yeah. And myself, I have been in the US for like over 20 years. And I work at mostly Novartis and BMS briefly. And I have worked on low molecular weight antibody and now, gene therapies. Yeah. So I guess I have seen a lot of different projects, Yeah. Okay. Yeah. Very nice to be here today.
Yarong.
Hi, everyone. This is Yarong from Guibao. Let me introduce myself a little bit. Actually, I got PhD in China, at University of Southern California, and I have a big passion with cell therapy with more than 15 years of CAR-T development in both US and China, and 2 of them CAR-T program are currently in BOA stage. So in 2019, I co-founded Guibao, tried to focus on developing tumor-infiltrating lymphocytes for solid tumor patients. With 4 years, the Guibao has established a lot of platform, and we have 3 generations of TIL products. Try to extend the indication other than melanoma and cervical cancer. And right now we have GT101, the most advanced TIL product in China. Currently, is in pivotal phase 2. We aim to file the BLA by the end of next year. Hopefully, we can deliver the first Chinese TIL product to Chinese patients soon. Thank you.
Thank you all. As we all know that we witnessed the really great success even this year. So far, we have witnessed the 4 gene and cell therapy products approval globally, and including the first TIL product, including the first gene editing product from Vertex and CRISPR Therapeutics. But for each individual niche area that you guys are focusing on, there's pros and cons, there are strengths and weaknesses. So I would like you guys to comment on in your area, what is the strength, what is weakness that we are currently experiencing for achieving great efficacy in various diseases? And maybe we can start from from Yarong.
Okay, I think for the TIL therapy, which is the Gribao is focusing on, the most challenging I can see is that actually it include, for example, the first one is how to manufacture to kind of get TIL product with high tumor specificity. It's really hard. For every TIL company. Try to normalize the manufacturing process to deliver the drug with the same quality. And the second one, I think, is that how we can extend the TIL therapy to other indications for other than melanoma and cervical, which has been shown kind of a good clinical outcome. And third one is even after infusion, including other, for example, the CAR T therapy, this kind of products still kind of face to the suppressive factors coming from the tumor microenvironment. In this case, how we can kind of do gene modification of the TIL product to kind of overcome this tumor microenvironment is a big challenge for this therapy.
Okay, one further question in terms of TIL compared to CAR T, which is very successful already, right? What is the advantage of TIL?
The advantage, I think the most advantage coming from the TIL is the TCR kind of the clone type. In each TIL product, actually, it contains a lot of TCR products that can target a lot of tumor neoantigen. So this one is cannot beat by CAR T or kind of TCR-T, this kind of mono target.
Thanks. Dr. Tian?
From what we have been working on, like both gene replacement, gene editing, I want to first focus on like a gene replacement therapies that we have been working on. It's all like AAV-based gene therapies, yeah. And for our projects, when you select AAVs, honestly, right now, like the AAVs that you could use, there are actually not that many different serotypes could be used. So my company, part of what we've been doing actually is focusing on identifying better AAV, which has like a better like a tissue tropism and efficacy, and hopefully, as a result, we could reduce the dose. Yeah. So that's like for viral-based delivery vehicles, yeah. And other things about like AV therapy is like endurance of AV therapy. I think that's like an issue. And so something like we also have been working on, looking for ways, whether, one will be like be able to dose patients with pre-existing antibody, and other is like how do you like increase the endurance of your therapies, and another thing is allow maybe second or third dosing, down the road when it's needed, yeah. And I would also like to talk a little bit about, we also use like a non-viral based delivery system like lipid nanoparticles, yeah. And for lipid nanoparticles, I think right now the biggest challenge, and a lot of companies, and also like in the US, like NIH and the Gates Foundation, they are spending a lot of money, has this big initiative is actually to identify and develop lipid nanoparticles which would allow targeting in vivo directly and target the tissue of interest. Because right now, most of the LNP used is mostly like a liver targeting. So it limits the kind of projects that you could take on. And you could imagine if you could identify like a LNP, which will allow you to target any tissue or cell type of interest, that would greatly expand the type of projects that you could take on and the type of disease that you could cure. Yeah.
I want to just follow up on the AAV and also gene editing part a little bit more. So for AAV side, as you mentioned, there's a lot of limitations for AAV as a vehicle, right? It's highly efficient, but not efficient enough to deliver to most of the organs. For example, liver, only 40-50% of efficiency. Muscles, about the same. Neurons is about 5%, right? How do you increase the delivery efficiency of AAV as a vehicle?
Yeah, I think that's a big undertaking. Actually, a lot of companies are doing. Trying to find more specific and more efficient AAV could deliver to the target of interest, right? Tissue of interest. Therefore, it will reduce toxicity, increase safety, and will also reduce the dosage that's needed, and the cost will be lower. And I think one of the problems that, because my company is also very actively trying to identify novel AAV capsids, which we would be able to use in some of our projects, more efficiently. And I think the problem is like a lot of times, the data you obtain from preclinical research, even like non-human primate studies, you never know when you go to clinic whether the translation is going to be there. Therefore, I think this is something which is quite challenging. And for people who are familiar with like AAV gene therapy, SanegeneBio, they actually owns like a lot of like AAV patents, like AAV9, and some of the earlier ones that people use. And there was one time we had a discussion with their team, and I noticed that even though they claim they have more than 100 different AAV capsids, but you look at all their projects, they actually use AAV9. Okay, so I asked, why don't you use, the novel capsid that you claim that you have in your pipeline? And the reason is that if it works, why do you want to change? So I think it's a balance. When do you so we constantly encounter the same problem because we also have identified some novel capsids. So which project and when would you be ready to use it? Because with novel AAV capsids, I think manufacture, CMC, it requires like a totally different optimization and validations, right? And so it's a lot of work compared to if you just use AAV9. Yeah, so I think the company will just have to take the plunge, You really believe in your capsid. And maybe sometimes you will have to do side-by-side comparison, maybe in small-scale IIT clinical trials, and to definitely demonstrate the advantage of the normal capsids, yeah.
Great. So one follow-up question in terms of gene editing side. Gene editing is a great tool, and we see the first product is being proved this year, but that's in vitro gene editing product. It's not in vivo. Looks like you guys are working on in vivo gene therapy side, in vivo gene editing side. And I think it's very difficult to increase the efficiency of gene editing, not only the delivery side, but also editing side. What is your comment on this? How do we overcome this efficiency problem?
Actually, our experience, at least with two of the projects that in vivo editing project that we have worked on, efficacy, at least in like a non-human primate models, that data that we have seen. And also like, in the US, there are quite a few companies also already have early like clinical stage in vivo gene editing projects. I don't think in vivo efficacy is a big issue. However, my concern has always been like, if you deliver, depends on how you deliver your CRISPR-Cas, right here, we are talking about CRISPR-Cas. If you use like a LNP to deliver, right, and because the CRISPR-Cas is not going to be around that long, therefore, I don't think I worry about the off-target effects, the long-term off-target effect as much. However, the problem is like, CRISPR-Cas is delivered by LNP, and as I mentioned earlier, that really limit the kind of target, the kind of disease you could work on. And I think right now that's really the rare limiting factor. If you see like most of the company or all the companies that in the US or in China that has like a in vivo editing project, they all target liver. Yeah, because we are limited by the delivery vehicles. And that's why I was saying earlier, it's very important for us to identify vehicles, will allow you to do in vivo projects and deliver to target of interest. Yeah, not just liver. Yeah.
Sounds great. Thank you so much. Now coming to Shiyu, as you guys work on RNA have seen tremendous success in clinic and commercially. What is the newest technology in RNA field that is working really well?
Thank you, Zhenhua. So talking about CGT, right? So RNA, small interfering RNA, actually right now, at least to my knowledge, as Zhenhua just mentioned, was considered as the most mature, actually, the subdivision of CGT now. So the reason for it is that's a subdivision of CGT, although that's a fully chemical synthesized molecule. So it's still small, it's kind of with a lot of chemistry entities there. So it's from the manufacturer, from the design perspective, it's a lot better controlled, especially from the CMC perspective. So, but for the mechanism of small interfering RNA, the discovery was win the Nobel Prize back to 2006. And then right after that, over 20 years effort, right now there's 6 FDA-approved drug, actually achieve very good potency and safety profile. So the reason for it is, first of all, it's the scientific mechanism is very solid. And then second of all is that the target, right? So because it's based on the specific disease-causing gene, and then to further reverse and modify the physiological and the pathological conditions. And then targetability is relatively accurate compared to the other modalities, other biologics. And then the last thing is that because it's a highly stabilized chemistry entity embedded into the molecule design, and the duration of the molecule can chill half year of 1 year right now at the moment. So, and then people used to say that the small interfering RNA, the therapeutics can be considered as a part of immunization, and then so that a year is basically is easily achievable goal in terms of duration of the modality. So different properties for the drug, and then can make the drug to be successful at this stage. But I believe there's a lot of going on. And then again, a small molecule type of categorized molecules. So the PK property is still not that ideal, especially for extrahepatic delivery platform. So along that way, there's still a lot of technology breakthrough need to be done, as Zhenhua just mentioned. And then, but everything else for the small interfering RNA would be delivery, and delivery.
Yeah, I totally agree. And as you know, RNAi has been around for many years and never didn't had a breakthrough until you guys discovered the GalNAc as a delivery tool. And GalNAc is only working really well for liver currently. And what is the new technology that can, for you guys, can go beyond liver?
That's a good question. And then, Zhenhua, so for the GalNAc hepatic delivery system, right? So the receptor and the GalNAc, the binding affinity is extremely strong. And also the receptor property at the hepatocyte and is very abundant and then with very fast recycle recovery rate. So basically, people used to say that this GalNAc SGPCR delivery system is nature designed. It's basically, it can be perfect than that. So whether the company, the field, and also Syngene are able to find out another delivery system to really reach the levels of hepatocyte delivery, and then as the drug already approved by FDA so far, we confident, but I think it could be a lot harder than GalNAc and then the current receptor system. But for Syngene and for the technology, we have been focusing on, because the liver is a bit different, right? So it's the major organ, and the 50% blood will circulate to that organ and then give automatic lot exposure to that organ, and then no wonder, the drug uptake and the efficacy is relatively better compared to other organs. So for Syngene, so we are trying to develop a platform, not only include the ligand as what we used to use GalNAc for the purpose, and we also include certain levels of PK enhancer, and to really add different properties of the drug molecule to increase drug PK property, and then also targetability for different cells and then organs, and hopefully we can reach very similar potent levels of the drug. I think it's definitely achievable because we have already got some very promising data from our NHP study for different cell and beyond the liver. I think it's not easy, but I think it's doable, at least for certain organ tissues like adipose, CNS, and muscles.
Go beyond your company, any other proof concept in clinical setting that you can go beyond the liver?
So right now, the one very good example coming from Alnylam, and so the CNS delivery system. So although they did intrathecal kind of local delivery, but the drug potency is quite good, and then the durability can reach close to a year. Yeah, it's quite good. Yeah.
Sounds good, but compared to RNAi versus ASO, antisense oligo, right?
Right.
That antisense oligo has already reached the CNS, has already achieved the proof of concept, has already achieved a market success. What do you compare to RNAi for CNS versus ASO for CNS?
Right. So the intrinsic property as a drug molecule, oligo and siRNA have quite big difference, because the oligo itself is a single strand and the siRNA is a double strand. And just from the structure perspective, small interfering RNA like siRNA is a lot more stable. So in terms of the durability, and the siRNA is a lot better, I would say, a lot robust compared to oligo. And on the other hand is that, again, that's all back to the chemistry structure. So, and then a certain modification inside the oligo can cause, sometimes can cause unpredicted toxicity profile that has been observed in different clinical programs so far. So I think from that perspective, I think we do believe that small interfering RNA definitely has certain advantage over oligo. So no matter CNS or hepatic programs.
Sounds great. Thank you so much. We spent more than half the time on the first question. So let's move on to the second, which is safety related, right? As we know, for CAR T, recently there's a lot of discussion in terms of oncogenesis for the CAR T, for the modified CAR T. And for RNAi and for gene therapy, there's also related safety concerns. So I want all the guests that comment on in your area, what is the safety concern and what is the solution to those safety concerns? Maybe let's start from Yarong again.
Okay, so for the TIL, I mean, the TIL field actually, the big kind of concern of the safety actually coming from the IL-2, the high dose IL-2 as a standard adjuvant for non-gene modified TIL product. Especially, I mean, even with the first approval and the approval of Antavi, the first TIL product in the world, they still use the standard, I mean, high dose IL-2 as an adjuvant with TIL therapy. In that case, I mean, for TIL therapy, we think further development should be kind of try to do some gene modification of the TIL products to decrease the dependency of the cytokine, especially the IL-2 in vivo, to enhance the safety of the patient.
Sounds good. Thank you so much. Maybe Dr. Tian?
I would like to focus on gene editing. Okay, for gene editing, I think there are a few aspects. First of all, increase the specificity and efficacy of your like CRISPR-Cas in this case, because my company, we are using Cas12b, and this is a CRISPR-Cas that have been optimized internally, and we believe that our Cas12b, we call Cas12b Max, okay, it's not only very efficient, as good as Cas9. We have done, we have collaboration and in addition, I think the biggest advantage from we have evaluated up to a dozen different targets side-by-side comparison with Cas9, and in all the not all, but the majority of the instances, what we have seen is less off target of our Cas12b, and so reduce off target effect. Okay, I think that's important, especially if you want to deliver your Cas by AAV. Okay, there are some projects that, like I mentioned earlier, you couldn't use LNP to deliver because the tissue you want to target, and therefore, if you use AAV to deliver, then you want off target frequency as low as possible. So that's one thing. Another thing would be I mentioned earlier, if you use LNP to deliver, then how do you optimize your LNP, and the supporting, material like the RNA optimization, etc. And therefore, you could use significantly lower dose. And lower dose, not only the cost will be lower, but in addition, I think off-target, frequency and effect will also be lower. Yeah. So I would definitely focus on, because I think at the end of the day, gene therapy, gene editing, especially in vivo gene editing, the biggest concern is off-target, and in the long term, the consequences of target effects,
Sounds great. Shiyu.
Okay, so talking about the small interfering RNA, Zhenhua mentioned, right? So hepatic platform and extrahepatic platform. So the safety profile and tolerability profile actually embedded in those platform. So delivery itself is a concern in terms of toxicity consideration. And on the other part, right? Because for the gene therapy, it's always sequence related. So the second toxicity consideration would be coming from the sequence off target effect. So for the delivery platform, in terms of GalNAc hepatic, it's approved. It's very, very safe. So because of the receptor, because of the ligand, and because of how we design the molecule. And then for the extrahepatic, depending on what ligand that has been using, and then clearly the safety profile not as good as a hepatic delivery platform, but the drug eventually can still reach the tolerable rate, as I just mentioned as example for the CNS platform that demonstrated in the clinical progress by Alnylam is pretty good. So, but for the sequence-based off-target effect, that's none of the platform really can avoid it. And then the company has tried different ways and then go above and beyond and then use a different chemical method and to try to minimize off-target effect, for really focusing on the C region of the payload and then kind of more or less how that problem solved. So for our couple clinical program, basically for the liver, especially for the liver, so we have really kind of a challenge the HP and then over 100-fold than the therapeutic dose in human. It's really, really safe.
Thank you so much. The next question will be focused on manufacturing cost. I think I'm going to skip Shiyu because luckily they don't have to worry about the manufacture cost as a synthetic way. For cell therapy and for gene therapy, manufacturing cost is still major concerns for the commercial side. What is your comment on that side, on that particular issue? Maybe start from Dr. Tian.
So I think what I mentioned earlier, like increase the efficiency and specificity of your tool. Like for example, AAV, and if you use like a LNP for delivery, you also increase, the LNP, efficiency, and therefore you could lower dose, and the manufacturer, burden will be lower and could also reduce the cost. Yeah, and obviously, dual manufacture, process, you can always streamline, and yeah.
Great, thank you. Yarong.
Actually the cost of CGT product is always a big concern to most people. But please note that actually, the product, especially the autologous cell therapy, is one-time short treatment. Compared to traditional big or small molecules. So to me, the overall cost of the CGT product actually is not that much different compared to the traditional therapy with multiple treatment cycles. That's the first thing I want to mention. And the second is that for most patients who receive the CGT product, the life cycle of that patient was much better compared to traditional therapy. So when, especially probably, later in the future, when CGT product can move into the earlier lines, for example, the first line, with the much better efficacy, and the patient can benefit more from kind of one-shot therapy with and benefit from the more quality of life. I think the cost of CGT can be justified. And also in China, with cooperation with local, for example, the equipment and reagent supplier, I believe the cost of CGT product can be further getting down, in the near future, and more patients can be probably can be benefit from this product soon.
Sounds good. I would also add a comment on gene therapy related products, for especially AAV gene therapy related product. In the beginning, something like Zhenhua, which is a great successful commercial product, the cost of Zhenhua is about 6 million RMB per patient, which is incredible, which is also unsustainable in China market or Asia market. And now with the process improvement, with the scale improvement, also with the newer regulatory improvement, the cost can be reduced down to 50,000 RMB per patient. So this is almost 10 times of improvement there. I think with the time goes on, this is going to be even better. For example, for ocular gene therapy product, we can now reduce the cost down to 100 RMB per patient, so which is really minimal. We have about 1 minute left. I would like all of you to really envision the next 5 years of gene therapy, gene and cell therapy research. We'll start from Shiyu this time.
Thank you, Zhenhua. So I think I really believe that small interferon can really go beyond the liver and then can reach other organs and to solve a lot of unmet medical needs.
Yeah, I think hopefully within the next 5 years, there will be much better delivery tools, allows to target different tissues more specifically and more efficiently, and therefore increase the safety, and reduce the cost of gene therapies.
Thank you. Yarong.
Hopefully, the tool can extend the indication to other, for example, the cold tumor, and more patients can get benefit from the tool therapy.
Thank you, everyone. Thank you for our guest.